Today I will talk about our Imalytics preclinical software. It's our main product of my company, Gremse IT, here in Aachen in Germany. Spin out from Fabian Kiesling's Institute in Aachen. And today I will talk about quantitative imaging, quality control, and dosimetry. And I'll give you a brief overview, like the scope. So you're all familiar with this pipeline here in research, where you have your animals like mice. You image them, you acquire raw data. These are micro-CT projections. And then a reconstruction is performed here, PET-CT, for example. And then you generate regions, you perform quantification, and then you have some results, hopefully significant or not, about your mice, your subjects. And the scope of the software that I'm presenting today here is around here, the image processing, segmentation, and quantification. And that's where the Imalytics preclinical software is useful. And for today's talk, I'll give a bit of a motivation. So there were recent successes with lutetium-177 PSMA therapy in the last years. Here you see a patient with lots of metastases. They are really being reduced by a few injections of this theranostic lutetium tracer. It's FDA approved. You can use it with a PET companion diagnostic, but you can also image the lutetium tracer directly with a SPECT. And this success has really stimulated a lot of research in this field to develop more tracers or more therapeutics like this. And so last year we took it up here, part of my team here with the Montreal group, and we decided to support this because we learned that there was not really good software solution, especially for the dosimetry. So especially Carolyn Anderson gave us a good talk and introduction about that. And so the workflow here that I'll also go through today as an example workflow is to load the SPECT-CT data with the 4 mice, for example, split up the data, adjust the metadata for quantitative analysis in %ID per ml, generate absorbed dose maps, segment tumors and organs, and then get absorbed dose curves, which can help you then for your therapy design. And so this is an example here with the software. I'll show it with the real software in a moment. So you can crop the data and split it into several mice. Then you adjust the metadata if it's not present in the DICOMs. And then you split it into multiple folders. So because the analysis is typically done per mouse. And like this, I'll show it in a moment. And then we go on with Monte Carlo dose point kernels, which we found here. There were more than 2,000 very accurate kernels published in medical physics, and we are using them to apply the dosimetry computation to go from the biodistribution image to a dose rate map. And then here this is GPU accelerated. And then we have the dosimetry curves. But I'll switch over now to the software so that you get an idea. So this is the software, and you can load in some image data just by dragging it in here, CT. So it's loaded here, CT of a mouse, and you can drag in a SPECT or PET or file, and then you have the software here. You can visualize it in 3D, open different slice views, and this is a lutetium scan. So the image quality is not great, it's quite blurry, but that's because it's lutetium and you don't have so many gammas for imaging. And then here the image data is in, we have a nice unit here. Sorry, I have the bar here. And Here it's an SUV, but it was already converted to SUV, but you can convert it to percent ID per ml here. And now you can, with our dosimetry tool, you can convert it to a dose map, here, a dose rate map, where you select the tracer, here, lutetium, for example, or any other tracer. You can enter the injection, how much you hypothesize to inject for your therapy. And then you click apply. Then the kernel is applied, which is very fast because it's GPU accelerated. Now you have the unit here in gray per second. And you can save this dose map. And then you can load a organ segmentation, which you or create one here. And then this would be done for each of these time points in the folders. Here for the mice, for this mouse. And with our batch mode, you can go through the folders here and inspect it. Now you can use the dosimetry tool to compute the cumulative dose. Here, but we need to go to the overlay. And you get here the analysis over the time points, a big table, and the dose rate profile here. So this is the curves of the dose rate in milligray per second for the different organs, the kidney, liver, lung, muscle, and tumor. And you see here that the liver is, for example, going down, the tumor is going up, and you have a bit also in the lung and in the muscle. And then what's interesting here is the total, the cumulative dose, and that is the area under the curve, under each curve here. And we compute this here and show it here. And you see we have 11 gray, for example, in the kidney, 8 in the liver, and here in the tumor. And if we zoom out a bit here, you can see that we extrapolate also into the future beyond your last scan time point by fitting a bi-exponential curve to each of these curves, because the dose continues even after your last scan time point. And this allows us to extrapolate it. And this is then the projected total dose here. So the estimate on what is eventually delivered to that organ. And then here you can see the cumulative dose. It's just a running integral for each of these curves. And here this is converted to ZVAT here, the effective dose profile. And with this tool, the scientists can assess their therapeutic potential with their tracers, and they don't need to necessarily go from, they don't need to do SPECT imaging with lutetium. They can also use a companion diagnostic. So you can determine the biodistribution with a pure PET tracer, and then with our software here, virtually switch the isotope and compute the absorbed radiation dose. Now I need to go back a bit because I forgot this at the beginning. I wanted to show this workflow with the 4-mouse bed. So before doing this analysis, in many cases, these mouse hotels are used here. This is a scan, a CT scan with 4 mice. This is for high throughput imaging. And then the data needs to get split. And you can do this with our software here really easy. You can jump to one mouse here, like this mouse, in 3D, because there you can see better where is left and right. And then here with the cropping tool, you can crop out one mouse like this, and this crops both the CT and head file. And now you can adjust the metadata here because in DICOM you currently cannot really store 4 injection doses for 4 mice, so you have to kind of get it in here another way. So you can fix it, enter the injection dose and the weight for the mouse here, set it, and then save the data as DICOM or as a different file format. And then you click undo and you go back, and then you can do the same for the next mouse here and so on. And this allows you to quickly sort your data on a mouse level, because then the analysis is typically done per mouse and not with these 4 mouse images. And if you want to make a figure for a publication, the reviewers would likely also want to see it per mouse and not in the full with the 4 mice. So if I go back here to the slides now, the dose maps are computed with a GPU acceleration here. And I'll give a brief overview here. So the nice feature from the modern PET and SPECT systems are that they provide you quantitative image data with a nice voxel unit in typically megabecquerels per ml or kilobecquerels per ml. And then this can be turned into biodistribution with a percent ID per ml voxel unit. And from this, you can go to a dose rate map in gray per second as a voxel unit. And there you have different, you need to do this for every time point because the mouse would be scanned like every day or every few hours. And then the question is, what do you do next? Because then you need to perform organ segmentation to associate your data to the organs. And I'll explain this a bit here. This is different from dynamic imaging. I'll show you an example here in our software. By the way, this is a tutorial button here in our software, and we have a lot of examples installed with the software. Here, the 4 mouse data, but also many CT applications, MRI, ultrasound, and our Python extension. And I'll show you one 4D data set here. So this is a dynamic FOG scan. And here we can go through the time and inspect the data here. And then you can generate a segmentation. I'll just give you an example here real quick with the ISO tool. Segment the heart here, or the bladder. Or here, let's go to the kidney cortex a bit earlier. The center. There are many more segmentation tools also with delineation. And then you can do the quantification. You get the organ curves for your organs. And so, but this is dynamic imaging. That is great. But with for the dosimetry, this is typically not done because you have longer time points. So you scan the mouse several times at different days here, first day, second day, third day, or after here 3 days. And then when you load the data, you will notice that the data here is a bit, there's another situation. So you can go through the time here with the batch mode. The problem is that the mouse is repositioned every time. It's kind of moving around, it's bent in a different way. You can try to position it similarly, but it's not like in a dynamic scan where you have one position over your whole 4D scan. It's multiple positions. And then you need to segment the organs here. Time point. Like this, I can go through the time points here. You see we have different organs, tumor, kidney, heart, liver, lung, and muscle. And then you can quantify the concentration or the uptake in each region. And then that is giving you the information over time, the curves. So if I go back to the presentation, here, the segmentation can be done with our software interactively. And we are working on an AI mode, which can take a lot of work from you. Then you don't have to do it manually or interactively. And then you finally have here your results. So you have the segmentation, and then you get these curves, the dose rate curves or cumulative curve to allow your prediction of your how much you can inject without killing the mouse based on the dose limiting organ. And we validated this dosimetry tool actually using clinical data because we found some nice data sets with this publication here. We didn't have that for mice, but our software also works for clinical data. It's just not approved for real diagnostic or therapy planning. And our results were in a similar range like to other dosimetry softwares used in the clinical scope. Here we got similar values. And we also noted that these softwares also have quite a difference in their understanding or in their results, because it can be quite application or implementation specific. And when you do this quantitative imaging, it's quite important to keep an eye out for the quality control. And in that regard, we have been working intensely with Fantech, a small spin-off company in Wisconsin in the USA. And the typical problem with quality control is that, I quote a scientist here, the scanning takes 5 minutes, but the analysis half a day, so we never do it. So we worked together and adjusted the phantoms. Here you can see it with little cone tips in 3D. Those are markers, holes. Just a moment when it's starting again here. You can see the holes here. And they are detected by our software. This is a Lorenzo phantom, automatically detected, used for image fusion. You just need to select the type of phantom, and then we compute these line profiles here and provide you the recovery coefficients, for example, and an estimate of the achieved resolution. And I'll show that here with the software. We have the data sets here in the tutorial section. Let's load this Lorenzo phantom here. And here you can visually see the resolution. And here in 3D, you can see the phantom here. It's around the size of a mouse. And there you can even see the serial number. And then you can perform the automated analysis here for the Lorenzo phantom. You need to select the one you have scanned, but you can see it here in 3D. So you select it, and now it's computing, and it found the cone tip markers, it performed the image fusion, put markers here, and generated the recovery coefficients for the different rod sizes here. So the big rods, 2 millimeter, you get 74% recovery, but for the small ones, here they are pretty blurred away, so there's almost no recovery. Here are the line profiles through these cylinders here. and a sigmoid fit on the function of these fractional recovery values. We have something similar for a partial volume correction phantom here. This is looks a bit different, the phantom. It's a bit longer, like a mouse roughly, and there we have the cone tips at the front at the end. And inside the phantom here, if I go through it with the clipping, you can see there are these spheres all connected, so it's easy to fill. And then I can perform the automated analysis, but first I need to convert to %ID. Here I can do this with this dialog, and here you see all the metadata is populated because we got it from the DICOM. So click apply, and now the image was converted to %ID per ml, and now you can run the automated partial volume correction phantom analysis. You need to select the proper one, and then here you get also the recovery coefficient. Here, see for the large spheres, it is quite high here, close to 1. For the small spheres or the empty sphere, you have a lot of loss of signal due to the intensity diffusion. And here we compute the recovery coefficients based on the %ID that we expect. So you can check if that matches. And here we use it relative to the largest sphere. We set that as 1. That's why you get a 1 here. And as a third example, I would like to briefly show you another pretty useful phantom here. It looks like a mouse in 3D, but it's a 3D printed phantom. And if I change the windowing, you can see that there are actually a lot of screws here. So you can fill this. I can go here to the website of Fantech. Here, this is the biomouse. And these are the other phantoms. And so this has 11 chambers. You can fill it with different amounts and concentrations of your tracer. And then you can perform the analysis here, the automated application pretty fast. And now you have the organs here. It's using the rings as markers, rubber rings. And now you can compute here the uptake per organ. Here in the mean concentration in kilobecquerel, or the total amount. And if you convert to %ID per ml, here you get the quantification in % of injected dose here. It says milli %ID, and then you see how much you have everywhere. And this, as I mentioned, this is quite important for your quality control, or quite useful to be able to check if everything is working properly, like your scanner, your image data, but also your software analysis, to validate that your whole processing actually makes sense. It's much easier to check it in a phantom than in a mouse, actually. And about the software, the Imalytics Preclinical software, it's available for Windows systems, Mac OS, or in the cloud, where we use Microsoft Azure Cloud. It works well on workstations or laptops. You can also use NVIDIA GPU. It's recommended for large data sets. And we offer also affordable group licenses. So it's really possible to have it installed on like basically any computer in your lab for a reasonable price. It's easy to learn. You can basically become productive in one day. We have a YouTube channel here with more than 74 tutorial videos, and a lot of tutorial datasets are installed with the software. And it's extensible. We have an open file format here published and disclosed, and we work this out with 3 device vendors and 3 academic institutions, and we provide free C++ and Python code to load and save our files. And we have a Python extension if you want to develop your own custom plugins for our software. And it's compatible with all devices here, anything with voxels or pixels, and we can read and write many file formats, DICOM, NIFTY, Analyze, even the old Siemens Invion files, if you have one of those systems. And it also works on MRI data, MR PET, ultrasound, MPI, or fluorescence, bioluminescence. And it's installed and used on more than 60 sites worldwide. Also pharma companies, and it has been used for many publications, more than 100 publications. Here are some example images here, and here are some more. And yeah, then I thank you for your attention, and I'd be happy to answer questions if there are any.